Hello everyone, all the way from Denmark. I would like to thank the organizers for giving me the opportunity to present at this virtual US UPO conference. My name is Dorte Becker Jensen. I work at the Danish company Evosep, and I would like to share some very recent data obtained with our new whisper methods. The Evosep One is a separation system delivering 5 different standard methods, covering a range of use cases from comprehensive proteome analysis with fractionation strategies to ultra-high throughput single-shot analysis. By using a disposable trap column, the so-called Evotip, peptides elude off at low pressure and store the preformed gradient in the loop. The high pressure pump then pushes the preformed gradient with separated peptides over the analytical column. Thereby we can deliver extremely robust flow rates over time, here shown by 150 and 25 nanoliter per minute. One of the hottest topics and the next big thing in proteomics is a growing field of single-cell proteomics. It requires outstanding sensitivity, but also throughput and robustness. Therefore, we developed a new set of methods directly to cover this growing need for ultra-high sensitivity analysis. We have 2 methods available, both operating at 100 nanoliter per minute, with a duration of either 30 or 60 minutes, leading to a throughput of 40 or 20 samples per day. The full width half max is around 5 seconds for both methods, and the retention time is robust, here visualized by monitoring 2 peptides over a course of 15 injections with both methods. It's kind of a catchy title, but nevertheless the truth. As said before, the Evotip serves as a disposable trap column, which integrates solution with liquid chromatography. Therefore, we avoid one of the steps implemented in a classical workflow, namely to elute and dry down the sample. In this process, you can also lose valuable sample. Furthermore, it serves as an excellent way to concentrate the sample. When pursuing ultra-high sensitivity, every step matters, and it's important to limit these to avoid sample losses. Samples are robustly stored on the Evotip and can be for up to 35 days, which is as long as we tested with 550 and 500 nanogram loads respectively. Initially, we compared our new 40 samples per day whisper method with our standard 60 samples per day method. For peptide elution, window is roughly the same, of around 20 minutes. Flow rate is a factor of 10 lower with the whisper method, running with 100 nanoliter per minute compared to 1 microliter per minute with the 60 samples per day method. Electrospray ionization is concentration dependent, and therefore we expect a decrease in signal from lowering the flow rate with a factor of 10. Indeed, we observe an increase of 100% in protein identifications with the whisper method compared to the standard method, leading to 1,500 proteins identified from only 1 nanogram of HeLa. This was analyzed with DIA on an Orbitrap Exploris 480 ms and searched with Spectronaut. This coverage from 1 nanogram diluted HeLa is extended to 2,000 proteins when extending the gradient to 60 minutes. We see a saturation effect with 40 samples per day, whereas the longer methods take advantage of higher loads. We observe high quantitative reproducibility, here plotted with CV percentages. A median CV of 10% is calculated across dilutions and replicated, and free replicates. For the super low load of 1 nanogram peptide, around 70% of the proteins are quantified with a CV percent below 20, which increases with higher loads. In addition to this, we would like to see if the increased sensitivity also benefits phosphoproteomics. Therefore, we again benchmarked our performance against the established 60 samples per day method. We performed independent enrichments down to as little as 1 microgram diluted HeLa peptide using our automated Kingfisher Flex platform and magnetic titanium IMAC beads. With a normal input range of 200 microgram, it was quite striking to see the excellent coverage with the whisper method of more than 1,000 phosphopeptides identified from 1 microgram in DDA mode, which is an increase of 300% compared to the 60 samples per day sample. The reproducibility was also really good, with a Pearson correlation of 0.98. We optimized our workflow even further and took advantage of the increased sampling in DIA mode. This increases the coverage another 150%, with almost 3,000 phosphopeptides routinely identified from only 1 microgram of peptide input. Reproducibility is still excellent, covering almost 2 more orders of magnitude compared to DDA at the 1 microgram input. This ultra-high sensitivity phosphoproteomics application opens the door for new and exciting things, such as tiny tissue samples and FFPE samples. We decided to test this further in C. elegans. One worm is around 1 millimeter and comprises 1,000 cells, representative of our optimized workflow at 1 microgram input. To minimize the sample preparation steps, we performed an in-solution digestion directly in the Kingfisher plate, which was then directly used for phospho enrichment. We were quite happy to see that we identified more than 2,000 phosphopeptides from a single worm and then reproducibly over 4 replicates. We identified a little more than 4,000 phosphopeptides from 2 worms and 6,500 from 8 worms. Phosphopeptide intensity increases as expected when analyzing more worms, as it's visualized to the right. The combined pool of phosphopeptides were clustered and visualized in a heat map analysis. From the single worms, ribosomal phosphoproteins are enriched, and from a cellular component enrichment analysis, we see that we cover phosphoproteins from the cell, such as cytosol, extracellular vesicles, ribosome, cytoplasm, and also the nucleus. Ultimately, we wanted to see if we can actually see differences in the phosphoproteome upon inducing stress, in this case by heat shock for 1 hour at 37 degrees. We analyzed 8 single worms, which was processed and analyzed individually, 4 control and 4 treated worms respectively. We were happy to see that we are able to separate the phosphoproteomes by a simple PSA analysis. We observed variation in the control samples, but this also highlights that it's 4 individual single worms analyzed, whereas the heat shock treatment seems to measure a more robust phosphoproteome. From a differential significance analysis, we observe regulation on the phosphosite level, even from a single worm analyzed, with ribosomal and heat shock phosphosites significantly regulated. In conclusion, I hope I convinced you that our new Whisper methods are ideal for ultra-high sensitivity work. They are plug and play using a standard hardware setup and standard columns and emitters. As an additional application to the true single-cell work from Andreas and Marvin in Matthias Mann's lab in Munich, they have a paper on bioRxiv. We have demonstrated the gain in sensitivity for phosphoproteomics applications with more than 3,000 phosphopeptides identified from 1 microgram of HeLa peptides and more than 2,000 phosphopeptides from a single C. elegans worm. With these methods, we can gain biological insight from tiny sample amounts to better understand disease. I would like to acknowledge the entire team at Evocep for pulling this off, and especially Nikolai and Ole. The phospho data was done by Florian, who is a PhD student in Jesper Olsen's lab, who I would also like to thank for an excellent collaboration. Besides that, I would like to thank our collaborators at Western Biosciences and the Max Planck Institute in Munich, and you for your attention. Thanks.